Now that we've discussed the events of the cell cycle in which the cell is growing and getting ready to divide, we'll now move on to talk about the process of mitosis. Mitosis is the process of dividing the nucleus in a eukaryotic cell. If you remember back, eukaryotic cells have multiple chromosomes, and it's a much more complex internal structure, so it's necessary to go through several steps in order to divide the cell successfully to make sure each daughter cell gets a complete copy of everything. The stages in mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. And then that is followed by, or actually it often overlaps telophase, that process of cytokinesis, which is separation of the cytoplasm, so that you end up with two daughter cells with pretty much exactly the same thing. The spindle is a structure in eukaryotic cells that is necessary to successfully divide the chromosomes into two equal and separate entities, two separate nuclei. It's composed of microtubules and is organized by an area in the cell called the centrosome. In the centrosome, this is the microtubule organizing center of the cell. In animal cells, it's centered around the centrioles, which are a pair of barrel-shaped microtubule structures that are found in animal cells. Plant cells also produce a spindle, they just don't have centrosomes to help organize it. Here we see some pictures of a cell going through the stages of mitosis. In interphase, of course, you can't really tell that anything is going on other than the cell is getting bigger. You can't see when the DNA is divided, but you can see sometimes the centrioles replicating and becoming separate bodies. And that's one of the last things that happens in interphase before the cell progresses into prophase. In prophase, you've got an early mitotic spindle developing here between the centrioles. As the centrioles or centrosomes migrate apart from each other, you can now see chromosomes within the nucleus. Oftentimes you can tell by looking at that they're duplicated, composed of two sister chromatids attached to the centromere. In prometaphase, which is kind of a transition step between the prophase and metaphase, you see that the nuclear envelope is fragmenting or breaking down. The special tubules that attach to the centrioles, called the kinetochores, are attached there, and you can see the microtubules of the spindle more fully developed than you saw before. So in interphase, once again, the contents of the cytoplasm have doubled. Okay, it forms two centrosomes, two microtubule organizing centers. The chromosomes get duplicated during the S phase, of course, and the nucleolus and other structures like that are visible inside the cell. This is a cell in interphase. You can see the microtubule organizing center there, and you can't really see any other details inside the cell at that time. So here we see pictures paired with the interphase, prophase, and prometaphase structures that are shown here in the diagrams. In prophase, the microtubules begin to emerge from the centrosomes, and the centrioles start to separate from each other, forming the spindle. In the nucleus, you can see the chromosomes have coiled up and become compact, and you notice that the nucleolus has disappeared. In this picture, you can definitely see the little string-like or thread-like structures that are the chromosomes. They're still not completely compactly tightened up, but you can see that it's happening there. In prometaphase, the microtubules from the spindle are reaching the chromosomes where they attach on the centromeres. And what they're doing in the process of prometaphase is gradually moving the chromosomes toward the center of the cell. The nuclear envelope disappears, and other structures are no longer visible except for the chromosomes and the spindle. Here's the cell in prometaphase, so you can definitely see the thread-like chromosomes attached to the spindle here, and they're moving around in the cell. In metaphase, the chromosomes are going to line up in the middle. Anaphase, they will separate from each other, and then telophase, they'll form two new nuclei. Cytokinesis produces two new daughter cells. And here we see the accompanying photographs to go with that. In metaphase, the spindle is fully formed now. We've got chromosomes lined up in the center. We call that the equator or the metaphase plate of the cell. And the kinetochores, or the attachment points of the chromatids, are facing opposite poles of the spindle. Here's a cell in metaphase, and you can see that the chromosomes are all lined up right there in the middle. the centrioles are lined up in the middle with the tails of the chromatids sticking out there. In anaphase, those centromeres basically separate or split from each other. Since the centromeres separate from each other or split apart, the chromosomes can now move to opposite sides of the cell. There are special motor proteins similar to what you find in your muscles that move the chromosomes along the spindle fibers toward the opposite ends of the cell. As this is happening, the cell gets longer because the other microtubules are kind of pushing against the ends of the cell, making the cell get bigger so that it's more easily divided. And here's a picture of the cell in anaphase. You can see definitely that the chromosomes are being pulled away from each other, and the cell is definitely getting longer. In telophase, the cell is going to continue to elongate, and basically what happens is the nucleus starts to form. The nuclear envelope starts to form around the chromosomes. The chromosomes start to uncoil. The nucleoli reappear, and the spindle goes away. And usually about the same time cytokinesis gets started. Here in this cell, in this picture, you can see that definitely we're forming two nuclei. You can see that the cell is starting to pinch in here, and you can see that the centromere spindle is starting to disappear here in the center of the cell. In cytokinesis, of course, we're going to divide the cytoplasm into separate cells. It works differently in plant cells and animal cells, and so we'll look at the process in each one. In animal cells, cytokinesis occurs as a cleavage furrow forms. The cleavage furrow is formed by a ring of microfilaments that tightens or constricts in the middle. It's kind of like a drawstring on a pair of sweatpants. When you pull the strings, it gets tighter and tighter, and that's kind of what happens in the cleavage furrow. As the cleavage furrow deepens, it's going to separate the contents into two cells. Kind of like if you used a string to cut through a piece of Play-Doh or something like that. Here we have you can definitely see the cleavage furrow here, and in the diagram it shows us that we've got this ring of microfilaments that is tightening and contracting, and eventually will cut the daughter cells into separate cells. In plant cells, it's a little bit different because remember, plants have a cell wall that has to form, and so a structure called a cell plate forms in the middle between the two daughter cells from vesicles that are made containing cell wall material. When the mitosis is complete, then those vesicles migrate to the center of the cell and start to fuse with each other, and the cell plate grows outward from the center all the way to the edges, and as it reaches the edges, it'll divide the contents into two cells, each with its own membrane and cell wall. Here you see a picture showing the cell wall forming, the cell plate there in the middle between the two nuclei that are reforming together with the chromosomes loosening up. There, the cell plate is forming in the middle. And in the diagram, you see that basically you have the vesicles there. They start fusing with each other and adding their material to each other, and you'll end up with a cell wall dividing the two cells.